Good afternoon and welcome to the second of the USDA Human Nutrition Research Center on Aging's Fall Monday Seminar Series. Before introducing today's speaker, I just wanted to mention that each month this year we will be featuring a speaker from the different HNRCAs, and today's speaker will be our first. It is my pleasure today to introduce Alexis Wood, who will speak on using metabolomic data to develop personalized nutrition approaches. Dr. Wood is an assistant professor at the USDA ARS Children's Nutrition Research Center at Baylor College of Medicine. Her research examines biological pathways relating dietary intake and health, and to investigate these relationships, Dr. Wood brings a breadth of training and experience in developmental psychology, psychometrics, statistical genetics, nutritional epidemiology, and applied biostatistics. Much of her work has used this integrated data from the lipidome, genome, epigenome, metabolome, and microbiome to link nutrition to cardiovascular disease and its risk factors such as obesity and type 2 diabetes. More recently, Dr. Wood has begun to investigate nutrition and health in children, applying the methodologies and techniques that she's used for her research in adults. Dr. Wood is investigating the interplay between nutrition and cognitive development in children. And with that, let me turn the screen over to Dr. Wood.
Great, thank you so much. That is a very spot-on summary of a winding career path. So I think you did a better job than I ever do in my bio sketch, so thank you. So thanks everyone for coming. I'm very excited to be here, or be here virtually. I thought because I am at a USDA center, like I'm assuming the majority of the audience are, I would introduce myself a little bit as a colleague, a potential colleague and collaborator. I know of all the USDA centers, I probably have the most overlap with the Center on Aging, and actually have a lot of colleagues there too and collaborators already. And that also provides some context, broader context for how I see the bigger picture of each little project of what I do. And then I'll illustrate 3 projects that use metabolomics data to link diet to health outcomes, try to pull that together and say how I looked across those, drew some broader conclusions, and how I'm trying to investigate those further in my current projects. So the background for context, obviously I'm at another USDA center. I didn't know if this was sort of mostly open to the Center on Aging faculty or Tufts broadly, but we're a number of centers. We're in the Plains area. I don't know how many centers there are across the US. I should probably ask our USDA man on the ground, but the Plains area is 10 states and there are 22 locations. And I joined in 2014. And since that, nearly a decade, which is horrible to think about, my research has basically said I've taken high dimensional data, and I usually define that as, genomics, epigenomics, metabolomics, lipidomics, anything you can put an omics on. Stayed away from phenomics so far, but all those kind of things to link nutrition to health or other risk factors to health. And it started way back when with my PhD, which is very different. I'm trained as a developmental psychologist. And the goal of my PhD research was to better understand cognitive performance differences between children with versus without ADHD, to understand how cognition might differ as a sort of feature of the condition. And so the analysis was to parse the overlap in cognitive differences and behavioral symptoms in ADHD into genetics versus environment. The idea was to get at how much were these cognitive differences a core part of the ADHD disorder or condition, as we may see by shared genes, and how much were these differences that we saw perhaps arising from the environment, perhaps arising from the challenges, perhaps arising from how individuals respond to children with ADHD. And so when you take this overall schematic, which I'll come back to, and it still summarize what I do today, it just looked a little bit like this. It was twin data. It's the start of the GWAS era, but it was genetic relatedness. And the overall conclusion across 3 years and many papers were cognitive deficits, particularly as related to executive function, were a core part of ADHD, And actually, probably the primary starting point, and many of the inattentive and hyperactive and impulsive behaviors we saw flowed from these cognitive differences. And in terms of, sometimes I think about like, how did I get where I am today? Why am I still using this paradigm? And I think a large part was how impactful this was in my early career. So that when I came into it, ADHD really was purely behavioral. It was diagnosed on inattentiveness and hyperactivity behaviors. And that's still to an extent sometimes true today. But if you're seeking a diagnosis through a neurologist or a more concrete, sort of a more informed diagnosis, we actually use those cognitive deficits and those cognitive differences and those aspects of cognition to diagnose ADHD today. And as I have ADHD, I was diagnosed doing exactly the same test that I used for my PhD data like a decade before, used it in my PhD, not knowing if it would even really pick up ADHD control differences. So it was just really cool to look back and see the impact of it and see the use of genetic information, not just for identifying genes and so on, but seeing that it can eventually end up in clinics, this approach. So then I moved to the US for my postdoc, and my goal was to develop a more sensitive measure of early type 2 diabetes using lipidomics. And the overall analysis approach was to pass the genetic influences underlying lipid subfractions done by lipidomics and insulin and glucose homeostasis into shared versus independent pathways. Again, the idea was that if I could show that shifts in lipoprotein subfractions predated type 2 diabetes and were an intrinsic part of the development, they could be used diagnostically to identify prediabetes. So we go back to that schematic. And it's easy to see how, although sometimes it looks like you went from developmental psychology to diabetes, you went from twins to lipidomics, there was a sort of logical pathway. And we did find that shifts in the relative distributions of lipoprotein particle size, and it's a little bit more complicated than that, but essentially mark the early type 2 diabetes state, especially in the postprandial state. And I still struggle with whether it was, impactful. It was funded, I gave talks, but ultimately, I think the conclusion of the community at the time was these lipoproteins are too expensive relative to the information we gain and the added predictive ability to impact practice. And, glucose and HbA1c and changes in insulin sensitivity do a darn good job, and this is never going to go anywhere. I don't think it's quite true. There's a little bit of a revival now, which I'll bring back, and it actually weirdly popped up a decade later in my research. But essentially, I did not feel this was as impactful as my developmental psychology research. Although maybe there was just less time, right, for it to go from research forward. So from there, I moved into nutrigenomics. And I think if I have any colleagues in the audience, this is probably where I met you and how we sort of started working on shared projects. And the goal was just to use, which I'm sure you're all familiar with, genome-wide genetic information to link diet to disease. And this can be differences in SNPs, it can be expression differences, it could be functional information. This was really, of course, the era where GWAS, genome-wide association studies, I think, exploded. We got into consortia doing these sort of mega analyses and really having the power to address some of these questions. And it was very exciting. And obviously the model is this, right? So again, we've gone from developmental psychology to diabetes to something broader in nutrition and health. I find like drawing an overall conclusion from that period of research a little challenging. I look across like multiple papers from all these consortia, and I'm like, sometimes we sort of identify genes that would link diet to health or moderate the association, but sometimes we don't. And I never felt like and I could be wrong, and I'd love to hear afterwards people going, hey, you left this research in a meaningful way in 2017, and we've really moved on since then. But I don't think we ever reached a paradigm, to me, where we could kind of predict this and use it. I say, okay, if you have a condition marked by X or defined by Y, then genetic information is going to tell us a lot about perhaps what you should eat or your lifestyle and how to prevent that. I think we're still in a to me, in a stage where we know that our genetic differences are going to be a key part of, or we suspect we know that our genetic differences are going to be a key part of why diet influences health differently in people, but we don't yet know how to operationalize that in a succinct way that goes to clinic. That's just my feeling. I still, and I'm very careful saying it to this audience, wonder how much closer in that paradigm we are to this goal of personalized or precision nutrition from the point of view of using genomics in that quest. So by 2016, I'd used GWAS, I'd used EWAS, I'd used lipidomics, all trying to link diet to health. I didn't really feel we'd got anywhere, and I didn't feel I'd got anywhere with my previous work in lipidomics and type 2 diabetes, which was not quite as nutrition related. And I was just feeling like, ugh, like, what am I doing? A little nihilistic, like, where am I going with all this? And so when someone said, hey, why don't you put metabolomics in there? I've got some metabolomics data, try that. Probably looking back, I feel like it was obvious that I would have said, no, I need to expand into something that I'm finding more fulfilling, but I didn't. I don't know if anyone's watched this film, but there's a point you go, bring it on. And that's kind of what I said. Okay, bring it on, I'll give it a go. So this leads to these 3 projects, I'm just going to take time, that use metabolomics to link diet and health. They're both interesting projects individually for what they might say about that diet factor and that health outcome. But there's also a broader picture that I think emerged from the first 3 forays into this. And this, you'll notice, is a fairly long period for just 3 projects. And I remind everyone about the pandemic and the disruption that caused. So we know that numerous aspects of diet predict chronic disease risk and chronic disease progression. I imagine it would be very hard to find someone at the Human Nutrition Research Center on Aging that says what you eat and your diet does not influence your health in a meaningful way. But a knowledge gap, particularly, again, it was 2016, were what the biochemicals were, biochemical pathways were, and perhaps bioactive compounds on those pathways linking diet to chronic disease. In my quest for using metabolomics to try to identify those compounds and pathways, was really hope that if we could find them, we might be able to tailor dietary advice to those, which aspects of the diet really produce those key health-preserving compounds. It might give us clues for therapeutic options. That, the simplest is maybe it would be a vitamin, maybe it would be an amino acid we could supplement with. And it would also provide, hopefully, an intermediate response variable in this quest for precision nutrition of, is someone responding to the diet we're giving them in a way that will help their health? If I put someone on the Mediterranean diet, and as a group on whole, I know some people's diabetes risk will go down. If I can identify the molecule mediating that, then can I assess someone fairly rapidly after they've been on this diet for 6-12 weeks and say, yes or no, you are getting the health benefit that I or your clinician feels you need from this dietary approach. So that was why I got into it. And it can be summarized as, we've got dietary intake and health. There was this link, and lying on that pathway some way and accounting for some of that pathway, I hope will be found in the metabolome. So you have always on the day you're giving a talk, you wake up with a sore throat. So the rationale for looking at metabolomics was that plasma metabolites would lie along the pathways from diet to chronic disease. So what are metabolomics? I know a lot of you know this, but it is the qualitative and quantitative study of all the small molecules in a biofluid. When we use plasma, those molecules reflect, in part, the effects of diet after the food has been digested, processed, and absorbed. There were, at the time I got into this, emerging plasma metabolite associations with diet. Many of you will be aware there are many more now. Many of you have done many more of them. And that there are also plasma metabolite associations with chronic disease, clinical phenotypes, clinical risk factors, such, everything from diabetes to fasting glucose to inflammation that we think might be a risk factor. So the first investigation that I initiated was could we use metabolomic information to understand whether, and if so, why, red meat intake was linked to increased inflammation? And the intake of red meat, particularly processed red meat, has been associated with the development of the risk of type 2 diabetes and cardiovascular disease. One theorized pathway linking red meat to these conditions was increased inflammation. However, these pathways, these associations, were not supported by some of the more recent observational studies, and particularly not supported by data from RCT's randomized controlled trials as a whole. And of course, there are lots of reasons for that. There's no clear answer as to which set of evidence is more robust necessarily. But as a whole, this pathway was still, or this association I felt was still in question. And one of the things popping out of the literature, this sort of comes later, is that a key moderator of whether we see an association between red meat intake and inflammation and or chronic disease risk is BMI. So we use data from the multi-ethnic study of atherosclerosis, known as MESA, which is a cross-sectional analysis of observational data. It's a fairly large number, 6,000 older adults. At baseline, I use baseline data. Dietary intake were assessed by food frequency questionnaire, so we can always bear in mind the limitations of that. We had plasma levels of 6 markers of inflammation: C-reactive protein, interleukin 2 and 6, fibrinogen, homocysteine, and TNF-alpha. And we had untargeted HNMR metabolomic features on a subsample of about 4,000 of them, who were just randomly selected from the baseline participants due to sort of funding costs only having enough for those. And these were collected as part of another project. So we did show that red meat showed the expected association with one mark of inflammation. I should be clear, surprisingly, this was actually the unprocessed red meat. We did not see any associations with the processed red meat. That's surprising to me. I suspect it's a feature of the food frequency questionnaire we used at the time and how well it was capturing that aspect of red meat intake. However, when we control for BMI, it's worth noting the association between unprocessed red meat and C-reactive protein was no longer significant. A metabolome-wide association study identified 2 peaks. Each spectral feature within the peak were annotated the same, which was great, which were inversely associated with red meat at metabolome-wide levels of significance. That was histidine and glutamine. But again, it's worth noting, when we controlled for BMI, the association with histidine and unprocessed red meat intake was no longer significant. We were just left with the glutamine. Both glutamine and histidine were noted in the literature for being anti-inflammatory molecules. We saw that in our data, not controlling for BMI. When we controlled for BMI, I think I better make sure these are labeled correctly, but anyway, when we controlled for BMI, it only remained with one molecule. So taken as a whole, as a sort of nutrition investigation, our analyses were unable to support a relationship between either processed or unprocessed meat and inflammation over and above any confounding by BMI. Because when we looked at just those associations without folding in the metabolomic data, we did not see any significant associations when we controlled for BMI. However, glutamine, I should say histidine, was a plasma correlate of lower unprocessed red meat intake, and this was associated with lower CRP levels. So the intake of unprocessed red meat was associated with lower levels of an amino acid that should lower inflammation. So then we do have a potential pathway between unprocessed red meat and inflammation when we use the metabolomic data that we don't see as a whole when we just look at the self-report diet data and the plasma markers of inflammation. Notably, unprocessed red meat, when controlling for BMI, explained 1% of the variance in C-reactive protein, but the 2 amino acids jointly explained about 10%. And when we only looked at, I think it was histidine that was significant, that was up at about 7 to 8%. So sort of some interesting findings there, but I don't think it's not clear where to go from here in terms of how we feel about the relationship, or what we might conclude about the relationship of unprocessed red meat to inflammation. We did potentially identify a metabolite that accounts for a pathway, but it's certainly not novel. I don't think it's going to set the world alight. It was an amino acid. Now, it was lower with red meat. We might typically think those will be increased with red meat intake because they provide amino acids, and this was not that. But when I looked in the literature to say, hey, I found these, it was easy to find evidence of these being linked to both metabolomic analysis of red meat itself and metabolomic analysis of people consuming red meat. And they had known anti-inflammatory properties. So we sort of demonstrated it, what I would say in vivo, but a pathway that was already there. So this is an ongoing second investigation, which looks at the association between a Mediterranean-style diet and cardiovascular disease. Uses very similar data, baseline data from the multi-ethnic study of atherosclerosis. But this time, obviously, the difference is rather than intake of one food group, we're looking at a more complex dietary pattern. The diet health association, you can see here, is pretty modest between baseline Mediterranean adherence to a Mediterranean-style diet at baseline and incident cardiovascular disease over an 18-year period. It's modest, but that's sort of what we'd expect to see. So when we conducted a metabolome-wide study of self-reported adherence to a Mediterranean-style diet, we got a lot of peaks. Each dot is a spectral feature, so there's obviously a lot of, you can guess, are going to be correlated spectral features, which we found. You can see there are these nice blocks of correlated features. So I did an unsupervised clustering analysis, and of all the sort of spectral features, based on this, I did machine learning, reduced it down, and then did a clustering analysis on the remaining spectral features that seem to show independent associations in an elastic net regression. And what we found is that these actually, from the clustering analysis, fit into these nice groups, these nice clusters, which largely shared annotations, which we'd expect. We'd expect features that represent the same molecules to cluster together. So I took a mean of the spectral features in each cluster to come up with like a cluster score. That's what I see. I do see a lot of other people using these data that way. I don't think that's the only way. Some people just pick the sentinel spectral feature, which is the strongest association within each subgroup defined by the clusters. I'm not sure about that, because I'm not sure what it means. If you pick the strongest, then not everyone has that. Does that really represent that molecule for everyone? I don't know. So I took a mean. And of course, as we'd expect, the mean of the mean score for each cluster, metabolites in each cluster, was strongly associated with a Mediterranean diet, which we'd expect because all the molecules in there, or all the spectral features, were associated with a Mediterranean-style diet. And that's how they ended up in that clustering analysis. Well, what we did then find is that for some of those clusters only, those were pretty strongly associated with incident cardiovascular disease over an 18-year period. So the conclusions are pending. I need to sort of work with some people with more biochemistry knowledge, perhaps biochemical nutrition, about interpreting the links between these, the significant molecules and cardiovascular disease, and where these molecules come from, how they arise from the diet and the digestion of the diet, what they might mean for cardiovascular disease and its risk factors. But broadly, we confirmed a pathway. I don't, again, look at this and think, wow, this is going to shock the nutrition community, and people are going to be saying, wow, you have to follow this up. It seems to sort of give a little bit more information to a pathway we know is there. It is interesting that at this point, I felt was the first time I really encountered the big data analytical problems that everyone who did genome-wide association studies struggled with. We're a stringent correction for multiple testing, for example. We're only getting the low-hanging fruit. What do we do about all these SNPs that are correlated? And that really popped up with these type of data. So that was interesting. But looked a bit like, I would say, the previous investigation with red meat inflammation in terms of the bigger picture. So then I did one more investigation. It looked into dietary patterns for other individual foods and dysglycemia. So we used 3 healthy diet patterns. We used a Mediterranean style diet, the dietary approaches to stop hypertension, a DASH diet, and adherence to the USDA guidelines for dietary guidelines for Americans. We used the 2015 version because that's when these data were collected, what was in place, and that adherence to that was measured by the Healthy Eating Index. This time I used a new cohort. It's the microbiome and insulin longitudinal evaluation study. It's known as MILES. The PI is Mark Gaddazi. It has self-reported dietary intake data on about 300 African-American and Caucasian older US adults. It's done at 3 time points over 18 months, but when I started these analyses, we only had baseline data. And what's nice about MILES, or why I chose this, why I was working with this study, is they have very finely phenotyped aspects of insulin homeostasis. They have an oral glucose tolerance test. So that's why the, in part, why the sample size is less. I mean, this is within an R01, whereas MESA was done within a contract, but we also had to, you know, Mark and his team at Wake Forest and Alan Bertoni had to get people in to go through a full OGTT. We also, this is the first time I used a different metabolomics assay. So rather than all the spectral features that are sort of annotated in bins and sort of more challenging to use like that, I used the global metabolomics panel by Metabolon that has known and unknown compounds that are each represented by a sort of discrete value. So all 3 diet patterns showed the expected modest associations with glycemia measures. It didn't really change when controlling for BMI, which was good, surprising, good. But I'd found that actually with the DASH diet in a previous analysis in MILES that didn't use metabolomic data. But you can see they're kind of modest. These are standardized parameter estimates. We then did a metabolome-wide study of the 3 diet patterns. We found that several metabolites from 5 classes were associated with each diet at a Bonferroni correction for the number of metabolites. So we had amino acids, we had carbohydrates, cofactors, lipids, xenobiotics. We would expect that given the complex or the very various different types of food, the diversity of the food that goes into a dietary pattern versus a single food. And something that's not a focus of the paper, but is fairly interesting, is the extent we see overlap. There is some interesting I noticed was there is no overlap in metabolite associations between DASH and an AMED, a Mediterranean style diet that isn't also shared with HEI scores. But the HEI has some overlap with DASH that is not shared with AMED, and the HEI has some overlap with AMED that's not shared with DASH. Then, of course, there are these 7 compounds in the middle that are associated with all 3 dietary patterns. And I did pull it apart when writing the manuscript, and I wouldn't say anything jumped out as meaningful. There were three molecules that were associated with all 3 diet patterns in my analysis, either at metabolome-wide levels of significance, which is 2.0 times 10 to the minus 5. One of them just sub-threshold. So it was still at 10 to the minus 5, but just didn't quite reach that 2.0 threshold. And had also been associated with the same diet pattern in the same direction, actually at metabolome-wide levels of significance in other analyses. So there are 3 molecules of potential interest there, but when I looked at the association of those 3 particularly with our glycemia measures, there was a little bit of a bust. They didn't really show anything. They weren't significant. What can I say? They were not associated with glycemia. So wasn't sure what to do with this information. You know, we had 55 metabolites, 83 associations across the various diets in terms of pulling out which aspects of the diet, which molecules really definitively associate with diabetes and glycemia, and can we look and make recommendations? It was a very complex, but what I did do to sort of distill down what can we get that's a take-home message is create metabolite summary scores. So I standardized all the metabolites, the values for all metabolites associated with at least one diet pattern. I reverse scored them so that it was always a positive associate, where necessary, a positive association with the diet, and I combined them into diet-specific metabolite summary scores. And what we saw with these was, with the exception of insulin secretion, for 5 out of 6 of our glycemia measures with insulin sensitivity and disposition index, obviously taken from the oral glucose tolerance test, but the others done at fasting, we saw very strong associations with these measures, which was interesting. So the broad conclusion was each of these 3 diet patterns show modest associations with health. They show strong associations because they reach metabolome-wide significance with specific patterns of metabolites. They are both diet-specific and more, metabolites that more represent a healthy diet. But of course, as we all know, aspects of a healthy diet overlap between the 3 diets. I think they all contain, the score all reflects to an extent lower red meat and higher nuts and legumes or something similar, and higher whole grains. So we would expect, if those aspects of the diet are leading to alter the metabolome, we'd expect to see that in all the diet patterns. But particularly, we did find those diet-related metabolite scores were associated with dysglycemia. So the sense of this is, I looked at this, I got into this to kind of identify critical molecules that, if you eat a DASH diet, molecule X goes up, and that prevents diabetes. And so we need to get everyone and give them molecule X and make sure that if they're eating DASH, that they get molecule X. But so far with just these 3, but I don't think this is unusual now. Much more of these sort of big metabolome-wide investigations of diet are popping up. I don't think it's unusual to say it's a kind of a limited impact on identifying novel molecules and kind of a limited impact of identifying novel pathways. I would say I didn't identify any novel pathways, but other research perhaps is hinting at that. And I think it's because basic nutritional epidemiology has gotten us pretty far. I don't know how many of you know our center director, Denny Beer, who's fabulous. I love him to death, but he's famously, if you know him, anti-nutritional epidemiology. We measured what people eat, we asked them, and we looked for associations. And one day I was talking to him and I was like, yeah, there's definite limitations. You can't argue there are some remarkably robust conclusions. We're pretty close to knowing some real stuff about what you should eat in terms of the role of perhaps unsaturated fats, the roles of whole grains and nuts and seeds versus trans fat. And he did say, I can't deny there is some remarkable concordance, but I'm very nervous of how this evidence was generated, which is fair. And my view is we've actually got quite far. I think, immense value in trying to fine tune that on what type of fat and how particularly does it work. But we know a lot, and I'm not sure my approach to using metabolites is really going to get us much further. And it sort of makes sense because I'm basing it off known diet health associations. No one really questions that a Mediterranean style diet or a DASH diet or adherence to the DGA is good for you, and it's going to prevent to an extent or lower your risk of chronic disease, even if it's a small amount. So I looked at it and that was the way I put it. I'm not sure these are really individually nutrition manuscripts. I'm not sure they're going to tell us what we should be eating, which was a bit meh. But I did have a shift in perspective when I looked across them. And the one surprising finding that consistently emerged was that the association of diet-related metabolites were orders of magnitude stronger than the associations of diet that led to those metabolites with the same health outcomes. And so you can see this here. This is across AMED diet investigation in the red meat. We just see the much greater effect size for the metabolite scores. These are the data in miles. In the dark bars, you've got the dietary intake score and its association with a measure of glycemia. In the lighter bars next to it, you've got the association of that diet metabolites with the same measure obviously. And what you can see in each case is there's just a big difference. Of course, insulin secretion was not significant, as I discussed, but these are just much stronger. There's a much bigger effect size. And I felt, to an extent, having been through this with 3 investigations, there was some robustness, some reproducibility to this. You know, it was different cohorts. Mesa and Miles are not only different sizes, different race ethnicities. They were collected. One was in 1999. One was still being collected, but kind of, I think, 2018 onwards. I'd used different metabolomic platforms. I defined diet at different levels, and we had different levels of outcome, from glycemia to a risk factor being inflammation, to a hard outcome being CVD. And the question was why? And there's a bunch of reasons, and I don't have an answer. Of course, there is going to be, to an extent, a reduction, or to a large extent, in measurement error when we assay someone's metabolites versus when we ask someone what they ate. Slightly in the metabolite summary scores, there were aggregate metabolites and the associations. You could see it like a genetic risk score, but we didn't see that across. I didn't take that approach across all investigations. There are potentially the effect of aggregate risk factors on any given metabolite. So if I have a diet-related metabolite, I don't know that it only comes from that aspect of diet I'm looking at, or only from the diet. Of course, I try to do general sweeping, you know, does this just represent a healthy lifestyle? So looked at associations with physical activity, with sort of generally a healthy diet. If I was looking at one food, I looked at association. You know, we controlled for socioeconomic status, but we also looked at associations. And I didn't see anything compelling, but I wasn't designing my investigation to look at that. I think what interested me most is that what metabolites capture is the effects of our food after digestion, absorption, and metabolism. And there are individual differences in those processes for all sorts of reasons. There are differences between individuals, and there are differences within a given individual across time. And so what they represent, what they sort of say is, we don't know how this diet is going to affect you physiologically. And if we can get at that piece, then we start to say, what are the physiological effects on health? So I always represent it this way, that we've got food intake, we've got health, and in the middle, we've got metabolites arising from food intake. Here, along with a reduction in measurement error, we have digestion, absorption, and metabolism. And here we have, these are the 2 sources of heterogeneity in diet health investigations that metabolites can capture or allow us to investigate. You know, number 1, do people get different metabolites from their diet? And number 2, do they use those molecules differently? Or do those molecules index something different in different individuals? If I get histidine, for some people, does it index a certain genetic resistance to higher inflammation, and others it does not, as a crude example. Now, we saw in the previous data that diet health associations are modest, diet metabolite associations are generally stronger, and diet metabolite health associations are generally strongest, which led me to believe it was probably this first part where the greatest heterogeneity was, because that would be an explanation for why we saw the different patterns of associations when we looked at the middle. We looked at the before piece and the diet to metabolites and the metabolites to health. So in my ongoing analyses, I wanted to look at possible heterogeneity in the association of dietary intake with the metabolites. I needed a food with individual differences in diet health associations to see could that be accounted for by difference in diet metabolite associations. Avocados were a great test food for this, to look at this paradigm, because they're a good source of monounsaturated fats. We think their associations with health largely are attributable to that. And the association of monounsaturated fats largely differs depending on your dysglycemia status. So I had a neat way to say, okay, is that because your dysglycemia status moderates the bioactive compounds associated with eating avocado? They were a great test fruit. The avocado nutrition board was also prepared to pay me to do these analyses. which helped with the selection of avocados. Although I did approach them to do this because it fitted my paradigm. So the first thing we showed was, did the associations between avocado and type 2 diabetes protection differ by dysglycemia status? Is this a heterogeneous association that we might look at the deviation of pathways with metabolites? Yes, it did. We found that avocado intake was associated with 3 highly correlated spectral features. They were not annotated as a known compound, but they shared the same chemical annotation, suggesting they all represent the same thing. And that compounds showed strong associations with type 2 diabetes risk and with incident type 2 diabetes. Then what we found was the association of avocado intake with the spectral feature of interest differed by dysglycemia. This is a hint. This isn't, actually this is published now, but it was definitely like let's look at these data. We may not have the power to test it, but we do see that the associations in those with normal glycemia were considerably stronger than those with untreated type 2 diabetes. In fact, we just don't really see evidence of it. And then we see a similar picture with the health outcomes. So at this point, I finally began to feel better about this line of research, and it's why it's now forming my main presentations, because it may be, if I've wanted for the past 7 years to get at precision nutrition and helping people identify what they should be eating for their particular health goals, but also their individual response to diet, maybe metabolites are the way forward. So how do we take this forward? Good, it is quick. One thing I thought I could do was investigate this avocado dysglycemia pathways for clues, right? Probably not going to do this because I don't know how far I'll take this on the priority list because they didn't renew their funding, but the approach will apply to other diets and other molecules, and to look at a multi-omics investigation. And the initial foray into this was very interesting. So I was using data from NHLBI's TopMed initiative. They have a big ongoing project with metabolomics, proteomics, transcriptomics, genomics. I spent a lot of time working with these groups, establishing this, and it's really a really great data source. And I put this in here, say if there's anyone out there who wants to start working with these data, please do reach out. I mean, I can't give you access to this data, but I can connect you with it and help you connect with the people who can. Let's be very clear about that. So I did a GWAS, genome-wide association study of that molecule that was associated with avocado intake and associated with protection from type 2 diabetes. And what we saw was this nice peak. It's a little spread out here, right? But this is the tail we like to see. And that peak represents a cluster of lipid metabolism genes. So it's interesting, right, that there appears to be a molecule associated with avocado intake that provides health and protection. This molecule is also, to an extent, under genetic control. And the genes that control it are involved in the metabolism of lipids. And if we jump back, I did not waste my time between 2009 and 2012. Lipid metabolism may be a key aspect of the development of type 2 diabetes. It may not be clinically useful, but I believe it is pathophysiologically what's going on. So it was sort of lends credence to the idea that this metabolome-wide approach picked up this molecule, and we can start to see, well, now I know why the association between avocado intake and this molecule might be different by dysglycemia status, because it seems to be in the lipid metabolism pathways, and we think we now know that dysglycemia is a condition of in part altered lipid metabolism. So again, when we're looking at precision nutrition and personalizing nutrition, the hint of the story, it is in no way definitive, is whether you should eat avocados or not depends on how you will metabolize the lipid aspect, on how you will use lipids, and you generally metabolize lipids, and that is different by glycemia status. Therefore, whether avocados should be eaten to promote health or protect health or prevent chronic disease may differ versus if you have type 2 diabetes versus if you have normal glycemia. So it came back. So in the future, thank you so much, Paul, for capturing this, is to take this, and I'm starting to look at this in cognitive development. I'm working with Tim Hughes at Wake Forest, who has this awesome research program looking at vascular contributions to incident dementia. I am not going to butcher his very successful and fascinating long-standing research, but essentially how subclinical atherosclerosis and arteriosclerosis may lead to cognitive decline and ultimately incident Alzheimer's and Alzheimer's disease-related disorders. We're going to see what we can do about using metabolomic, working with him to look at how that, what that might tell us about what and if diet may prevent cognitive decline in older adults. And also, given the strong associations, right, of diet with subclinical athero and arteriosclerosis, how that might fit into dietary approaches with a personalized or precision nutrition paradigm, if possible, might prevent incident dementia. And for Chris, assuming most of you are familiar with the Chris USDA projects, I'm looking to do this also in children. Obviously not cognitive decline, but looking at cognitive development. And I feel like it's come this like nice full circle, right? I started out with cognitive deficits in ADHD. I ended up using metabolomics to link diet to health and diabetes. And somewhere in the middle is bringing that into cognitive development, both at the end of the lifespan, representing that interest in diabetes and cardiovascular disease, but back in early childhood, representing those my initial sort of passion, I guess, for developmental psychology. So that's the picture. Always I left time for questions, and there's plenty of thank yous to say. I would say through Mesa, David Harrington, everyone in Mesa, very much grateful to a huge team, but particularly David Harrington and Jerry Rotter, who spent a lot of time sharing their data, talking me through it, helping me interpret and know where to go with some sometimes quite challenging data. Thank you to Mark, who's the PI of Miles, of course, to the participants in these studies. I don't want to downplay Miles, but Mesa participants who've been participating for 18 years is an immense commitment. But even Miles, who've been participating through a pandemic, and all the funding I received. And I'm happy to take questions.
Okay, thank you so much for that really fascinating presentation. There's one question. Before people populate the questions, I just had a quick question. I was really very interested in your metabolite-based diet pattern scores. And obviously, you showed them to be very strongly associated with glycemia. Have you do you plan on looking at any other cardiovascular risk factors in the Miles study?
That's a good question. My instinct is no, but I don't have a justification for why. Miles is focused on the goal of Miles is to look at the extent the microbiome influence the conversion of prediabetes to diabetes. So I think that's the outlook of that group. But I probably should, and makes me think that as I'm writing up this cardiovascular disease story in Mesa, maybe there's some replication there that would be really helpful to pursue. So
a second quick question about Miles. You suggested that this is going to be a longitudinal study. So they're planning on assessing the metabolomics again in the same population?
Yes, and so unfortunately, or maybe fortunately for Mark, Mesa also have metabolomics now, the metabolome panel at baseline, exam 5, exam 6. And at both baseline and exam 5, they also have the same food frequency questionnaire, give or take some supplemental questions that changed. And I do plan to look at that longitudinally. What I'm planning to do, but please, anyone if you have a better idea, what I have approval to do, please let me know, is to basically do a multi-level model with time as a variable to identify the diet metabolite associations that remain the same across time, that significantly differ according to time, and use that as a starting point to understand diet metabolite associations a little better. I'm sure there's some statistical mess that'll sort out, but perhaps if we look at why they might change, because your population arranging. If I see these metabolites as giving me a window into how you're personally digesting and absorbing food, I can well imagine over from 1 to 5 is about 10 years that changes. So that's the plan, yes, to use longitudinal data.
Okay, so there's some questions starting to populate. The first is from Jose Ordovas. He first of all thanks you, says nice talk as always. And he says, in terms of the relation between lipids and glucose, what is the chicken and what is the egg?
Yeah. Let me think for a minute. I may think for a minute and have no answer. So you're right. I think that's a good framing of it, because I looked at lipids and lipoproteins and dysglycemia looking towards diabetes, and which was the chicken and which was the egg. I don't know. I think that's, if you'd like to give thoughts on that, I welcome that. Otherwise, that's something I need to take back and think about how to investigate that, because I really came into this just looking at lipids precede type 2 diabetes, but not looking at the dysglycemia. And maybe that's why clinically they'll just never be as informative, because if the dysglycemia comes first, then that's where we're going to look to predict and understand. But I think even if the chicken and the egg, to me, I guess, has never been a super important question, because sometimes it doesn't matter for me, for my approach, the temporal ordering. It matters that we know this is a problem and this is going on, and we fold into our understanding of how to conceptualize diabetes, treat it, how to recognize it, how to feed people with it, if it's going hand in hand. I think that's the answer. I'm sorry if that wasn't very succinct.
Okay, next question. Again, Zhifan Wang says, great presentation. Can you share how the metabolomic summary score was generated?
Yeah, sure. So for each diet pattern, I took each metabolite that showed metabolome-wide significant association with that diet pattern. So a floor of it, right, is if metabolite X was significantly associated with AMED and was just sub-threshold with DASH, it did not go into the DASH score. That was an analytic decision, but I think it's important to know when you think about it. And so for each of those metabolites reaching significance with a given diet pattern, the values for that were standardized. The values were reverse scored if the association was negative, and then they were just summed across all. It was sum, not mean, it wouldn't make a difference. In short, if this is what you're asking, they were not weighted. They were not weighted metabolite summary scores. They were unweighted. And some of the people I'm working with now are not very happy about that. And I think what I will do as I move forward in the next stage with longitudinal data is examine the performance of weighted versus unweighted metabolite summary scores. And I will say, I didn't create, a lot of people ask this, and I just wanted to get this manuscript off my table, off my desk in the end. A lot of people really wanted a metabolite summary score that represented the commonalities between the diets. And a lot of people assumed it was going to be about that. I didn't do that, but I might do in the future if enough people yell at me.
Okay, and next question is from Shandong Wang. In your 3 diet metabolite health investigations, did you find any association with glutamine as you showed in your first two investigations? And then this goes on, could you comment on how to target glutamine in cardiovascular disease? Goes on to say, it's long with almost, glutamine mitigates some of the risk factors for cardiovascular, such as hyperlipidemia, glucose intolerance, obesity, and diabetes, but excessive shunting of glutamine to the Krebs cycle can precipitate aberrant responses towards harmful metabolic pathways that worsen cardiovascular health. Okay.
So I think glutamine is one of the ones that comes up in the AMED CVD association as an inverse association with AMED diet and a association with cardiovascular disease health. So, or no, a positive association. I'm sorry. No, I don't do interventions. I'm a horrible epidemiologist who goes, here you go, now someone go and do the hard work. I will bear in mind very much what you said about that, because I I guess what I heard from you is there may be benefit, maybe up to a certain level, but you've got to be careful. You can't just fill people with this. And I think that's really helpful to bear in mind that it's never going to be simple, right? And don't oversell what you're doing, and put it in, remember to always put it in the bigger picture. That's how I defend epidemiology, right? What I'll say to Denny is, well, no one's going to draw a dang clinical practice out in associations. It's a bigger picture. And so that's helpful. Thank you. But no, I have no idea about how we would use it in practicality.
Okay. The next a question is from Chao Lai, and he's, again, thank you for your wonderful presentation. Do you expect dietary pattern association metabolites differ between populations in then parentheses ancestries? If that is true, should metabolites score be applied useful to different populations?
Oh, hi, Chao. Thank you for all your nice papers you've written and put me on. I don't know, but yes, I would assume that. I mean, if I'm looking at my argument with the final investigation ongoing, avocado and so on, is that lipid metabolism may influence the diet metabolite association when it comes to avocado in this thing. And obviously we've got things like the FADS gene cluster that already moderate the association of lipid intake with cardiovascular disease risk. So yes, you're right. And I think that's just super helpful because as I mentioned, this longitudinal data in MESA, and everyone just does the metabolomics across race ethnicity controlling. And I think you're really reminding me to break that down by race ethnicity, even if they don't have the power to do it within the full metabolome-weight association, to at least look at those associations. I do. Short answer, yes, I think race ethnicity moderates some, but not all of the diet metabolite associations. And yes, I am resolved to consider that more seriously in future investigations.
Next question is from Greg Dolinkowski. He asks, do you plan to identify the important unknown compound in your avocado study? And if so, how would you plan to do this?
Kinda. So ideally we do analytic chemistry. That costs a lot. So I'm not doing that. However, the people we have the spectral features on, we also have all the metabolomic and proteomic data. So I do plan to look at if there is a molecule in the various panels from the Broad that could help me identify that. That's how I plan to do it. I don't have a better way to do it without a chunk of money, and I welcome suggestions on that front.
And the last question, given the time, in fact, it's the last question. It says from Madeline Camo, thanks for the great presentation. Can you clarify what definition was used for unprocessed versus processed meat? Yes.
I am, because I'm chair of the nutrition committee and I do know the FFQs. So unprocessed included lamb shanks, steak, beef steak. Processed and ground beef, and processed included sausages, bacon, chicharrones, and that. Does that answer your question? Those were the foods that went into each group. And deli meats also went into processed.
I think that's the end of the question. So again, I'd like to thank you for a wonderful presentation today. And
thank you. Thanks, everyone. Great questions. Thank you.
Thank you.